So good evening, ladies and gentlemen. So I'll be speaking on NAFLD and focusing on insulin resistance as a part of the pathogenesis, and in alcohol more on the metabolism specifically. So the framework is the overview on pathogenesis of NASH, the role of insulin resistance in NASH, and how do we measure insulin resistance. In alcohol, basically more a focus on alcohol metabolism and how it adds to the pathogenesis of ALD, and whether these will have any therapeutic opportunities for NASH and ALD, and how can we exploit the similarities and differences in the pathogenesis. If we look at the source of free fatty acids, it can either be from the 60% of them come from the adipose tissue, 15% comes from the chylomicrons of the dietary free fatty acids, and about 25% comes from de novo lipogenesis. So 3 ways of the fat to come in and 3 ways to go out. The 3 ways to go out are free fatty acid oxidation, triglyceride storage, and third is exporting as VLDL. So 3 in, 3 out. So how does NAFLD occur? I really don't know. Maybe many more might be more sure. We aren't exactly sure. We've had the 2-hit hypothesis, which was proposed by Day and James in 1998. And subsequently there was the multiple parallel hit hypothesis, which was proposed in 2010. So NAFLD goes through, fatty liver disease goes through simple steatosis, NASH, and cirrhosis. The first hit which was proposed was insulin resistance, which was associated with sedentary lifestyle, high fat diet, and obesity. And the second hit could be oxidative stress, mitochondrial dysfunction, gut dysbiosis, and inflammatory cytokines. Now we know that probably these things all tend to happen together, and the multiple hit parallel hit hypothesis was proposed, where people with predisposed genotype like the PNPLA3 genotypes would be more at risk. The many factors would act together, which would include oxidative stress, gut nutrient derived signals, and insulin resistance as one of them. And I'll be speaking specifically on the insulin resistance. When there's esterification to triglycerides, the fat in the liver which we actually see is probably protective. And if you look at the free fatty acids, when they come into the liver, they may get stored as triglycerides, but the toxic metabolites are probably the metabolic intermediates like the ceramides and the diacylglycerol. The ceramides and the diacylglycerol. The overnutrition, excess of diet with fat and sugars has probably been considered to be a starting point in the pathogenesis of NASH. And among fat and sugars, fructose is probably the one which is the most responsible. Unlike glucose, fructose cannot be utilized by all the cells. The only place fructose can be metabolized is in the liver. So it's like a little missile which goes straight and hits at the liver. So fructose may not be the best thing for people who are going to get fatty acid liver disease. Now when we compare this role of fructose, I'll come back to what happens with the alcohol. So the free fatty acids themselves could be hepatotoxic. So free fatty acids would come from circulating fatty acids. From insulin resistance will cause lipolysis of the adipose tissue. There will be circulating free fatty acids will go to the liver. There will also be de novo lipogenesis, and all these will be broken down. The primary site of breakdown is the mitochondria. The broader arrow is the mitochondrial breakdown. You can also have breakdown in the P450 and beta oxidation in the peroxisomes. All this will result in release of reactive oxygen species and lipotoxicity. Insulin resistance was introduced in 1922. The first time the term insulin resistance was used was to describe diabetic patients who received large doses of insulin to control the hyperglycemia. Now, these were all insulins which were from animals, and there were a lot of antibodies which were produced. These were impure insulins, and that was the reason for this insulin resistance. We see less of that now, and insulin resistance is defined as a subnormal biological response to normal insulin concentration. The whole aim of insulin is to promote energy storage. There's the important thing about insulin resistance is there's a dichotomy in the liver. There's a dissociation of the liver on what happens to glucose metabolism and what happens to lipid metabolisms. So although the action on glucose metabolism is effective, the effect on the lipid is not affected. So the hepatic de novo lipogenesis, which is the storage, because it's promoting storage, is paradoxically increased. So what the pancreatic beta cells will do is they'll compensate by increasing insulin production till they get exhausted, and we will get diabetes. So the insulin resistance will happen both at the systemic level as well as in the liver. In the systemic level, there's an inability of insulin to lower the blood glucose levels due to the disrupted translocation of the GLUT4, the receptors, to the membrane of the muscle cells. So this results in decreased uptake of glucose by the muscle cells, and you will get hyperglycemia. In the liver, the insulin will have similar negative effect on glucose metabolism, in that insulin normally mediates the suppression of gluconeogenesis. Now that would be disturbed, but the lipogenesis continues to be preserved. So insulin is promoting insulin energy storage, and it's this is the adipose tissue, and this is the liver. In the adipose tissue and in the muscle, the GLUT4 receptors are there, glucose transporters 4 receptors, which will increase glucose uptake and break down the fatty acids. In the liver, it will promote storage in lipogenesis and glycogen synthesis. When you have insulin resistance, what is happening is all the effects on the fatty acids are disturbed. But when you look at the liver, the fat metabolism is not really affected. So this difference in effect on glucose and lipid metabolism is because the pathway diverts beyond the receptor. So the pathway by which the insulin modulates glucose levels has to diverge from the path that mediates lipogenesis downstream from the insulin receptors. And I'll show you a few cartoons to sort of make this a little better. This is the insulin receptor. So it'll act on iron. When insulin binds to the insulin receptor, it goes through a cascade of actions where it will stimulate AKT, and AKT will act on SRBP, EBP1C, which will promote lipogenesis. Now this is not getting affected. What insulin also does is blocks the FOXO1 receptor, which causes apoptosis, lipogenesis, gluconeogenesis. So the effect which is happening is here. So because this pathway is patent from here, but this pathway is affected. So when you have insulin resistance, the problem is here. So the negative effect which would happen with FOXO1 is lost. Now when insulin binds to the receptor, it normally causes tyrosine phosphorylation, which will go ahead and act on the GLUT4. This is a glucose transporter receptors, which are normally in vesicles and get transported to the membrane. But instead of, when you have resistance, insulin resistance, this tyrosine becomes serine phosphorylation, and that's how the effect on the glucose metabolism is lost. So I go back and show you, we go back to this pathway. If you look at it, so this when it's acting, the problem is happening at this level. So the effect on FOXO1 is affected. So the gluconeogenesis, the effect on glucose metabolism is affected. But in insulin, this pathway is continuing to be all right. So lipogenesis will continue. So you will have fat accumulation occurring in the liver. So insulin resistance in patients with NASH, it is obviously a key factor in development of NASH, and it's a consequence of increased flux of free fatty acids to the liver. And insulin will also promote, resistance will also promote adipose tissue dysfunction and altered adipokines and inflammatory cytokines in the patient. So this is a crude pathway of fatty liver where you have behavioral deviance and increased maybe leptin deficiency, excess of nutrients, obesity, and insulin resistance and fatty liver leading to complications. Now this is a two-way process where insulin resistance can cause fatty liver, and fatty liver itself will cause insulin resistance. So these are the cartoons which are summarizing the effect of NASH, where you have free fatty acids. This is the adipose tissue. We have the free fatty acids going to the liver, and we have the chylomicrons which are going, also adding to the free fatty acids. The third pathway of fat coming into the liver is de novo lipogenesis. This can get either esterified as lipid droplets, which are nice and good because they don't harm the liver too much, or they can undergo oxidation in the mitochondria and the peroxisomes. The triglycerides are either stored in the liver or exported as VLDLs. So one of the key things which happens is insulin resistance, which causes defective insulin signaling. There is, as a net result, increased reactive oxygen species, which results in inflammation, fibrosis, and apoptosis. And then there is mitochondrial dysfunction, endothelium ER stress, and adipose tissue dysfunction leading to adipokines. Dysbiosis acting in the gut also contributes, leading to an inflammatory step. And finally, fatty liver. Now the interrelationship between insulin resistance and NAFLD, both things have been shown. There is data to show that accumulation of specific lipids lead to hepatic insulin resistance, and also evidence to the contrary that insulin resistance also is a driver of progression of NASH. So obviously the question will come, which comes first, the chicken or the egg? But if you look, both are acting, it's one circle. You have steatosis causing insulin resistance, which will cause hyperinsulinemia, increased lipogenesis and fatty acids. So it's just the chicken and egg story. So both are happening in tandem to each other. Steatosis, insulin resistance, increased insulin levels, lipogenesis, fatty acids, steatosis. Now, how do we measure insulin resistance? That's the second part. The gold standard was hyperinsulinemic euglycemic clamp technique. And the other techniques which have been used are HOMA, which is the most widely used, and oral GGT, and a few more. Now, hyperinsulinemic euglycemic clamp, this was actually the gold standard. What they would do is they would give a standard insulin infusion, and as the sugar would fall, they would keep checking the blood sugar levels and keep giving 20% IV glucose. And the insulin sensitivity was determined by the rate of glucose infusion during the last 30 minutes. This is a 2-hour test. So you give insulin infusion for 2 hours, you check the blood sugars, and you modulate the amount of glucose which is required. So if you need a very high level of glucose, more than 7.5, it is insulin sensitive. If you needed low levels, this is resistant to insulin action. But it's a very complicated test. There's risk of hypoglycemia, and you would rarely perform in clinical care. So what would you do in clinical practice? You'd probably check HOMA. The others are the QUIC, which is the quantitative insulin sensitivity check technique. The absolute insulin levels. So if you have a value of more than 20 with normal blood sugar levels, it shows that there is hyperinsulinemia and insulin resistance. Also, there's ADIPO-IR, which is adipose insulin resistance, which is a multiple of insulin and free fatty acid levels. Now this is what we use. So those are all more techniques which, although the quick has been used, most of the people would be using HOMA, and the clamp techniques are only for experimental studies. This is based on the principle that the degree of fasting hyperglycemia is determined by a combination of beta cell deficiency and insulin resistance. So this will counter for people with, say, having chronic pancreatitis. So HOMA IR will be insulin into glucose divided by 22.5. And since the factor is, this is in millimoles, since the factor of conversion is 18, so if you look at in milligram percent, it will be divided by 4 or 5. Levels more than 1.8 to 2 would suggest insulin resistance. So this is not part of my study, but since insulin resistance is important part of the pathogenesis, many drugs like insulin sensitizers would be used in management of NASH. The second part is alcohol metabolism. So, okay, let me just stop here, and if there are any questions in this part before I go further, I'll take a breath.
You asked them what is the mechanism of insulin resistance, because that is the driver for all the pathogenesis. So what do you understand by insulin resistance? Ask them a question. What exactly is the mechanism we explained about insulin resistance? Because that is major driver of type 2 diabetes mellitus, atherosclerosis, fatty liver disease, all vascular accidents, right, predominantly associated with insulin resistance. What do you mean by insulin resistance? And now just Pankaj told you in details, quite through cartoons, if you would have observed, you would have understood.
Anybody can volunteer?
Yeah.
NAFLD, whenever there is lipotoxicity and adiposopathy, there is increased release of the TNF and free fatty acids, which binds to receptors and subsequently causes the serine phosphorylation of the insulin receptor substrates instead of the tyrosine phosphorylation. So it prevents the further activation of SREBP1 and inhibition of FOX, thereby altering the metabolic pathways.
You just remember, but do you understand what it is? Anybody?
What is the physiological effect? That's what I'm asking. In the periphery and in the liver.
That is very important. You see, insulin is as a explained tyrosine kinase receptor, which is a transmembrane receptor on the cell. Insulin receptors are present on which cells?
Set of cells, they said.
They are present in muscle, they are present in fat, they are present in lower cell. Predominantly. There are other cells also which can use insulin. Okay? And the receptor is a tyrosine kinase receptor. So this is the receptor, insulin is a ligand, it binds it. So when it binds it, it does something. One thing you know, it controls blood glucose. So simple, he showed you GLUT4 vesicles. So when it binds it, it sends downstream signals to push the GLUT4 into the membrane. It is like a vesicle has a hole like this. It fuses with the membrane and makes the hole. So the glucose enters. And glucose enters and is metabolized by TCA, tricarboxylic acid, right? Where acetates are produced. Extra acetate can become ketoside and fatty acid. He's told that insulin other action is to produce fatty acid. So when it cannot send the downstream signal, the downstream phosphokinase A and B pushes the glucose. When it sits and cannot send, the GLUT4 cannot go. So the glucose cannot enter to get burned. Right? So despite presence of insulin, the glucose cannot enter to the cell. So the blood glucose level will go. Right? So this is a situation where there is insulin, but glucose cannot be met. Right? Now this phosphate is always critical among every cells. Right? And it is activated by ATP, adenosine triphosphate. You remember this, right? Now what he told is the second important part. Direct lipotoxicity is a condition. He told multi-hit hypotoxicity, right? And he told in this condition there are increased fatty acid, because initially when you have a sedentary lifestyle, right? You have heard that many of the extra substances will get converted to fatty acids, that is de novo fatty acid synthesis, right? As well as storage in the liver. Now, one of the metabolites of the fatty acid is a diacylglycerol. So, he told diacylglycerol in the cell does phosphorylation of other enzymes like tyrosine insulin receptor. So, he was trying to tell you about serine threonine kinase upregulation, which takes away the phosphate because of increased fatty acid. Once it takes away the phosphate, insulin, despite sitting, cannot get phosphorylated, right? And cannot push glucose. Am I clear? Second, when insulin level goes up, what is its major action on biochemistry days? It stimulates lipoprotein lipase, endothelial lipoprotein, which increases fatty acid, mobilizes to the liver cell, because fatty acid receptors are on the liver cell, where he told they are oxidized in the mitochondria, beta oxidation, and it is overwhelmed. It is paroxysmal oxidations or cytochrome P450, which liberates free radicals, right, which causes liver damage. Second, he told about gut microbiome, right, inflammatory cytokines, and direct lipotoxicity. Is this clear? That's what he told. Am I right?
Thank you, sir. Thank you for summarizing.
So, are you clear? This is very important.
Why the fat in the liver and steatosis in itself is not so toxic. They are all triglycerides. But it is the beta oxidation and the free fatty radicals which cause the inflammation, which becomes the driving force for ongoing inflammation and fibrosis. That's why when we have steatosis with inflammation, then steatosis with inflammation and fibrosis. So then that's the starting of the progression of the whole disease.
He also told the insulin has two other downstream signals. He told AKT-mTOR pathway. You see, what is the mTOR inhibitors drugs which you use? Huh? Why you use it? As an immunosuppressant. So what it does actually? Actually, it downregulates your cell proliferation, sends transcriptor factors which do not allow cell to replicate in mitotic phase. So mTOR actually sends down signals for the cell proliferation, right? Cell replication, JAK-STAT, right? Therefore, insulin is trophic. So therefore, if you tie the portal vein, liver atrophies because insulin doesn't go, right? Therefore, high insulin level cause cell replications. And therefore, mTOR inhibitor you use also as an immunosuppressant, right? So this is the second part. The third part is SREBP, steroid receptor binding protein upregulation, which causes esterification of triglycerides and storage. So remember three arms. One is proliferation pathway, one is GLUT4 pathway, including lipid synthesis pathway. Right, in periphery drives lipoprotein lipase into the liver. ये चार याद रखोगे, four things, and hence insulin resistance causes all these problems associated with inflammatory cytokine because of cell damage, secondary because Kupffer cells are there. When there is a damp, Kupffer cell will get sense it, produce cytokines, plus gut microbiome, endotoxins, which will also stimulate HSC and Kupffer cells, right? Plus direct lipotoxicity. Is it clear, the pathogenesis? That's what you told?
There was a song by Harry Belafonte which said it was clear as mud but it covered the ground. So it's a little bit like that, but it's covering the ground. There's lots which we don't know, but really what Dr. Acharya has summarized is the crux. And the resistance in the liver level is not happening at the receptor. It's happening below the receptor. And that is why one part of the pathway is being affected and the other part of the pathway is being spared. The effect on glucose is affected and the effect on lipid is spared. And as it is, the insulin levels are higher. So that is why there's more lipogenesis. So I'll go to the...
A simple way to remember what Dr. Puri has told you, insulin resistance majorly is muscle and adipose tissue, partially in the liver.
Since
So
it's partially in the liver, that is why it's worse in the liver.
that is why the worst affected is liver. So you have high fatty acids in the blood because there is insulin resistance. All these fatty acids go to liver. Liver will sense it as the fat is not affected. The fat pathway is not affected. So it keeps on storing more and more. More fat storage, more metabolism, more toxicity.
So I'll go to the second part of the talk, and that is on alcohol metabolism. So alcohol has a first pass metabolism in the stomach as well. And some of the alcohol which we ingest doesn't go into the body but is oxidized in the stomach by the alcohol dehydrogenase in the stomach. And how much alcohol gets metabolized in the stomach itself depends on whether we are fasting or not fasting. So if you are in the fasting state, the alcohol will rapidly go into the duodenum and will not really get metabolized. But when you have a food, you will have a higher blood alcohol concentration in in the fasting state. So the first pass metabolism is low in alcoholics, especially in women, and they have decreased ADH activity and increased sensitivity to alcohol and higher blood alcohol concentration in women than in men after equal amounts of alcohol. So less than 10% of alcohol is excreted in the breath, in the sweat, or in the urine, and 90% is oxidized. And most of this oxidation is occurring in the liver, and there are no feedback mechanisms by which we can really change the significant rate of alcohol metabolism. How much alcohol can be metabolized? You want to go for you say you've had 2 drinks, how long can you wait before your alcohol levels go down and you can drive? So a person can metabolize about 170 to 240 grams of alcohol per day in an average 70 kg man, which is about 7 grams per hour, which is about 1 drink per hour. Many factors can modify this alcohol elimination rate. It's higher in women, it's small decline with aging. It depends on races and ADH, alcohol dehydrogenase isoforms. It's increased in the fed state, and fructose. Fructose also increases the alcohol metabolism. So if you have your vodka with fruit juice, probably you'll metabolize it faster. Heavy drinking causes increased metabolism, and advanced liver disease will cause decreased metabolism. There are 3 main classic pathways in the hepatocytes of ethanol metabolism, which all lead to production of acetaldehyde. First is alcohol dehydrogenase, which is in the cytosol. And if you remember, most of the fat metabolism was occurring in the mitochondria. The alcohol is mainly metabolized in the cytosol, some amount by the catalase in the peroxisomes and the cytochrome P450. The catalase has an insignificant role, and the P450 has a role only when you have a higher amount of alcohol intake. The other pathways of metabolic pathways for alcohol are conjugation reactions with glucuronic acid to form ethyl glucuronide. This may have interest in people when we want to look whether there's an alcoholic who has relapsed. But you will check their blood alcohol levels and they'll be normal. But you may find this ethyl glucuronide in their body fluids, in their hair, in the tissue, in the sweat for much longer than the blood levels fall down. And this may be a marker of detection of relapse in alcoholics. Similarly, fatty acyl esters may also serve as markers for alcohol intake after alcohol is no longer detectable in the blood. What is the effect of alcohol? Once the excess alcohol is converted, it has 3 effects. 1, it causes acetaldehyde production. Second, it will cause a change in the redox ratio of the NAD plus and the NADH. Thirdly, it will cause mitochondrial damage and lead to decreased ATP production. Alcohol itself can directly, other than the metabolic part, can also damage the phospholipids and stimulate the Kupffer cells and cause oxidative stress directly as well. One is through the acetaldehyde and second is directly. What does the acetaldehyde do? Acetaldehyde will bind to the microtubules, which will cause intracellular retention of normally excreted proteins and cause hepatocyte swelling. Again, increased production of reactive oxygen species, and this NADH will cause mitochondrial oxidation and reactive oxygen species. So direct action of alcohol and toxicity because of the acetaldehyde, which will cause hepatocyte swelling, reactive oxygen species, and mitochondrial damage. There are many types of alcohol dehydrogenase isoforms, and the class 1 is the most important. The class 2 matters when you have really high alcohol levels, and this is going to also decide what is the rate of metabolism of alcohol. This is the overall alcohol metabolism, and basically it's going to be oxidized. Alcohol will be oxidized to acetaldehyde along with NAD+. So you are going to utilize NAD+, you're going to change the redox potential, and this acetaldehyde will be converted by acetaldehyde dehydrogenase to acetate, which will subsequently be, like all other food, degenerated and metabolized to fatty acids, carbon dioxide, ketone bodies, and cholesterol. But in this process of utilization of NAD+ and formation of NADH, you are changing the redox potential. So this NAD plus NADH redox ratio is reduced, and this affects many reactions which would normally occur, like glycolysis, the citric acid cycle, the pyruvate dehydrogenase, and the gluconeogenesis. All these will also be affected. To regenerate this NADH from the reoxidize the NADH, you need shuttles, because this will happen in the mitochondria, and the intact mitochondria doesn't allow NADH to go through. So you have a couple of shuttle mechanisms which will translate this, what you're finding is NADH, and you want this back, you want your NAD plus back. So you need to go through shuttle mechanisms. These are the two shuttle mechanisms which you will use to transfer this to the mitochondria, where the redox can be corrected again. So the pathogenesis of alcoholic liver disease is direct toxicity, acetaldehyde toxicity, changing the microbiome by the alcohol, and the impaired intestinal integrity and barrier function also have a role to play. This will cause leakage of lipopolysaccharides, activation of Kupffer cells, and sustained inflammatory responses. So some differences between ASH and NASH, which we looked at as to what would be there. Most of the factors will be there. You'll have oxidative stress, you'll have pro-inflammatory cytokines, but what will be the gross difference? There are a lot more pro-inflammatory cytokines in alcohol. The adiponectin levels will be higher. The oxidative stress is much higher in NASH. And what you're having in alcohol is actually malnutrition, undernutrition, while in NASH you're having overnutrition. The dietary cofactors matter. In alcohol, it will be unsaturated fatty acids, which will be probably worse. In fatty liver, the saturated fatty acids will be important. So what it does is it changes the mechanisms of the targeted treatment. There are certain common pathogenic mechanisms which include the hepatocyte lipid metabolism, endothelium ER stress, apoptosis, immune activation, but lots of things are different between the two. Both have fat, but there's still certain differences. There's a differential pathway of the TLR4 in between ASH and NASH. There is difference in the insulin resistance, which is the most important part of NASH, is not there in alcohol. There is also dysregulation of different microRNAs in both NASH and ASH. So while there is fat, the pathogen is in both. There are cytokines in both, there's oxidative stress in both. There are differences in the way these have been achieved. So these are probably my final slides. So what is conserved? There is the altered lipid metabolism is there. The cell death pathways are there in both. The ER and mitochondrial stress is there in both. The innate immune responses are there in both, and fibrogenesis is occurring in both. So what is different is the microbiome changes are different in both these patients, in NASH as well as ASH. So studies have shown different type of microbiome changes. There is evidence of insulin resistance leading to lipotoxicity in NASH, but the role of free fatty acids and lipotoxicity has not been shown to be significant in alcohol. And certain differences in microRNAs. So I was trying to digest this article on therapeutic opportunities based on similarities and differences in alcohol as well versus fatty liver disease. And it looked interesting, but a little difficult to digest as to ongoing studies and how they are going, which were in two tables. The first is the therapeutic opportunities based on the similarities, and the second one on differences. And this is, I think, something when we go on to treatment at a later stage. To conclude, ladies and gentlemen, insulin resistance represents the most frequent trigger of fatty liver disease, and fat accumulation results from increased free fatty acids, but as insulin levels increase, de novo lipogenesis is also increased, and there's a dissociation of effect on glucose and lipid metabolism. As far as the alcohol metabolism, the alcohol dehydrogenase is the major pathway, and the major byproduct is acetaldehyde, which will cause damage. You need substrate shuttles and mitochondrial respiratory chain to regenerate the NAD from NADH. And there are both similarities and differences in the pathogenesis of NASH and ASH, which may have some therapeutic implications in the future. Thank you. So if there are any questions?
Ask him questions. I will ask you a question from the voice.
Yes.
If there is increased fatty acid synthesis in the alcohol, why there is no insulin resistance? And second, both alcoholic liver disease and fatty liver disease can coexist.
Both alcoholic liver disease and fatty liver disease can coexist. Yes, they can.
Yeah, clinical phenotypes.
They can.
So
lots of things are different.
is there any study of playing the pathogenetic interaction between insulin resistance and alcohol toxicity together?
No, I haven't read that. So if you have seen that, I'd love to know.
Okay. But why alcoholics do not have insulin resistance? Has anybody studied?
So whatever studies are there so far have not shown any role of insulin resistance in fatty liver disease. Most of it is toxicity because of the metabolites.
Yeah.
And insulin resistance is the prime mover of the pathogenesis of fat. Yeah. While toxic metabolites of alcohol are the prime movers of the fat accumulation in fatty liver disease.
That's right. You see, the very interesting thing is the fatty liver disease, because of lifestyle modification, occurs because of a secondary messenger, that is hyperinsulinemia and inability insulin to work. Whereas in alcohol, it is the direct toxicity of alcohol, as he explained. This is the polymorphism of alcoholic dehydrogenase and acetaldehyde dehydrogenase, and the redox potential, essentially, and direct toxicity, plus the gut microbiome, right? You understand? But alcoholic can itself increase fatty acid, right? Increase fatty acid storage. Therefore, you find the first stage is the alcoholic fatty liver disease, right? And recently, people are seeing both can coexist, and there is synergism, right? So many alcoholics will have associated insulin resistance, and it's important clinically, because if you see today in day-to-day clinical practice, you can find the whole phenotypic shift of the presentation has occurred, which has to be documented. Previously, you used to see liver disease who were malnutrited. You are now seeing sarcopenia, relative sarcopenia, who are obese. And you see obese alcoholics now, right? If you ask Kausal, Manav, Niraj, even Pankaj now, sure, in the populations, you will find obese alcoholics, in whom both are probably working. It's a select group of patients where both are working together. But the pathogenetic interaction probably we don't know. So there's interesting questions for many of the young people actually.
Something to understand from you. When you have these free fatty acids, they tend to go and get deposited as triglycerides in the liver. That's the fat that we actually see. But that's the fat which is not causing the problem. The fat which is getting beta oxidized and there's an oxidative stress is the one which is actually causing the problem.
True.
So the natural driver for free fatty acids into the liver, when it comes into the liver, does it predominantly take the triglyceride pathway? Is that the easy pathway for it to take?
These free fatty acids, like the DAG and the ceramides, are part of the metabolism which will result in formation of triglycerides. They're intermediaries in the cycle. And those are the ones which are once it is stored, it's not an issue.
It's a mitochondrial effect. Beta oxidation is a critical state. Right? It cannot take care of the large amount of fatty acids. That's one problem. Second, alternative pathway like lipid peroxidation or paroxysmal beta oxidation or mitochondria, also they are compensatory mechanism or additional factors. When that gets overwhelmed, because you see large amount of fatty acid oxidation will have large amount of lipid. That is one part. But remaining fatty acid will be esterified. Right? And will be stored in that. Because that is the basic defense mechanism. Otherwise your cell will go. So essentially your survival questions will come. So basic defense mechanism, additional things will be stored. That is same thing in fatty adipose tissue also. Right. The problem that adipose tissue is also not available in every individual. Particularly subcutaneous adipose tissue is not available in individuals. Now subcutaneous adipose tissue probably studies has not been done besides PNPL. I think subcutaneous adipose tissue also genetically derived. Therefore Asians do not have subcutaneous adipose tissue. If you see Manav or Pankaj or Kausal or me, we are thin fellows. If you become fat after taking food, our tummy will only come up. We don't have adipose tissue under our skin and under our other parts of the body. Right? Brown fat. No, this is a subcutaneous adipose tissue. Brown fat is different. Brown fat is a metabolically active fat cells, which has particular distribution people are describing, particularly in neck or in the axilla. That's a different thing. And brown fat increases if you are exposed to cold. Right. So interesting. That again has evolutionary drive. So since we do not have adequate subcutaneous adipose tissue, we do not have storage space. Right. So as Asians, you are more prone to develop increased visceral fat. So that's why you read imbalance between SAT and VAT occurs in most of the patients with fatty liver disease. The reason is this. Therefore, obese people, if you will be surprised, good obese people actually are protected. I think if you clinically very carefully if you see it, it is morbid severe obesity which causes problem. But obese people who has a lot of circulated adipose tissue, you'll be surprised. Many of them will have normal lipid profile or normal insulin without a diabetes mellitus. So this is very interesting because many factors are associated. So very difficult to clear, as Pankaj already told, multiple things. So this is a very fascinating area, and there are many factors, not one group.
One more just quick question about the hepatocyte ballooning. You said the acetaldehyde causes microtubular damage and that may have a role in the ballooning, because ballooning is an important histological feature of
That is all.
NASH as well as alcoholic liver disease. So
Sure.
I just wanted to know, is that the dominant mechanism?
No,
for
that's
ballooning?
one of the mechanisms.
One of them.
So thank you. Yeah. These are all transcription factors and nuclear receptors which are playing a role. Yeah. Well, I've focused only on more on the insulin resistance as a part of the pathway.
Sir, about the de novo lipogenesis, So does the insulin resistance influence that also? Do you know lipogenesis or what?
See, what is happening is once you have once you have insulin resistance, you are not able to control the blood sugar. As a net result, the sugar levels are going higher and higher. I'll just go back to that slide, and it's a nice slide which sort of, I'll go back to the slide. So once you have an insulin resistance, you're not able to control the blood sugar. So the beta cells are producing more and more insulin, and this pathway of lipogenesis is not blocked. The resistance is not occurring at the receptor level, it's occurring at this level. So you're having a dichotomy on the sugar pathway. Normally, the insulin should be able to control blood sugar. But on the other hand, this pathway through the SRBP1C is open. And that's why the resistance is not for this. The resistance is only for sugar, and you're having more and more insulin, so you're having more and more lipogenesis.
One more thing I wanted to ask you in this particular slide, it's a very good slide. In this, does the gluconeogenesis decrease when there's insulin resistance?
See, what insulin will normally do is block this. So if you are not blocking this, then you are having gluconeogenesis. This is a normal effect of insulin to block this. FOXO1.
In a normal individual, you eat food, blood sugar levels go up, insulin levels go up. It inhibits gluconeogenesis because there is enough glucose available already. More glucose, more free fatty acids, more conversion into triglycerides. So that's a normal mechanism. Now, the conversion of glucose and free fatty acids to triglyceride is not affected in a NAFLD patient due to insulin resistance. So that thing keeps on going on. So the effect of insulin resistance in liver is different as it is in muscle and in fat. So muscles take insulin, they sense insulin resistance, they don't take up glucose. Fat senses insulin resistance, it keeps on throwing free fatty acids into circulation, but liver does not sense insulin resistance the way it should. So for liver, high sugar levels are there. More and more of this sugar, more and more of the free fatty acid which are coming from adipose tissue are going into the triglyceride pathway. So more fatty liver.
Basically, glucose gets stored as fat in the presence of insulin resistance.
I'll stop here. Thank you.